So today is the first lecture of our course. So I would like to welcome everyone. And first of all, I want to thank you for joining the course. And other thing is that you have to complete all the requirements to get the certificate. So for the certificate requirements, we're gonna publish it on our page soon in our Discord channel. So you will get the rules there and what you have to follow along this course and how to do the assignments. So let's start with our first video. So in this video, we're gonna just talk about briefly the introduction of the databases which we're gonna use along this course and which are necessary to do all the later on analysis. Okay, so we're gonna discuss first about the protein databases, and then we can also discuss the small compound databases. So first I want to tell you that why we need these databases to do the drug docking, because these databases contain the protein information as well as the ligands or the drugs or whatever you want to try to dock with the proteins, and then see that if that drug can inhibit the function of the protein or activate protein. Okay, but first of all, doing that, you need to confirm that the drug you are selecting or the molecule you are selecting either gonna bind with the protein or not. Okay, so to do that, we have this database where we're gonna take the information, and then we along the course we're gonna teach you that how these database can be useful to do the in silico drug designing and the docking and other studies. So let's start. First, I want to tell you about PDB database, which is called RCSB PDB database. So in this database, you usually find the protein sequences. Okay, so there will be like these proteins. Okay, and the structure of the protein. For example, in this example, there is a 9x2W. So these are just IDs and the codes of the protein. You don't have to care about for now, but later on we will see that how these are useful and how you can define in your results these proteins. Okay, so this database is actually contain all the protein information that you need for your docking purposes. Okay, so for example, here I go to the search bar and I try to find one gene which is very famous in the cancer. So we can write T53, and here you can see that there are many variants come for just one gene. So you have to select according to your goals. So in this case, for example, I want this one, okay, T53 binding protein. So my interest is in this protein. Later on, how you find this protein, we're gonna tell you that how to select the proteins by the data analysis, but right now I'm just giving you the small introduction. So here, for example, you say that you want this protein, you write your protein name, okay, and then there are many results. So the thing is that you have to carefully look at which protein is of your interest. Okay, so let's look at the first example. For example, I wrote I need the T53 protein. So now here we have the the results, and here we have protein. Okay, so in this protein we have some information which is necessary for our docking purposes. So first of all, you have to see that which organism you are doing research. So for example, if you are doing on mouse, murine, or any other, or they are bacterial or viruses, so you have to see that this protein is like belong to Homo sapiens. So these results were taken from Homo sapien. So this could be our interesting, because since we are studying cancer in humans, so we need always the human proteins as our first priority. Okay, so here and this is the molecule. Here is the protein chain actually, and we are, suppose we are interested in this protein, so we just click here So now you have some data regarding the protein. So this panel is showing you that how your proteins are like strong, or how they are like presented in this database. Okay, either the scores are very good. If these scores are high, that means that your protein is more reliable for prediction. Means that author who published this study have this level of confidence. Okay, but of course each of this metric means different. So in our later studies, when we're gonna select our proteins, we're gonna discover what these matrices means, because right now we are just covering the introduction part. So we're gonna focus just on the big things for now, and then we go for small details. So in this protein, you see that we have this kind of structure. You can explore this structure. So we have 3D structures, we have sequence also, electron density, and all these things, and ligand interactions. So for example, in this study, maybe the author wanted to use some small molecule to dock with these proteins. So if you want to see which ligand they use, so this is the sequence of the protein, here you can see, and here is our ligand, where the author just did the docking part here. So you can see that there is a ligand and interacting with the protein part here. And this is all the ligand, and this is the protein part. So if you zoom out, you will find this is the ligand. For example, they used SO4. I mean, I don't know what it is, but when we explore it, we're going to find it what it is. So this is just first example. So we go back, and we saw that this protein contains a ligand, and this protein have chains. Let's see how many chains we have in our structure. We have chain 1, like chain A, which is from the Homo sapien, okay? And this is the wild sequence, so there is no mutation. Like this can be found in the normal individuals, not in the diseased or mutated, okay? Unless there are specified mutations, okay? So this is the chain A, and this is the length of the sequence contains from Homo sapien, and 0 mutation, okay? Then you see that there are more other chains, which is different chains. For example, chain B, chain D, also from the Homo sapiens, and there is a 0 mutation in that case. So this is also a wild type, okay, from the Homo sapien. So it actually depends that what you want to study, okay. So later on we will filter these chains, and we just take our interesting chain. Okay? Also, this protein have a chain Z, okay, from Homo sapien, and no mutation again. And the ligand they used in this study was this one, sulfate ions, okay, and with the structure this, and with zinc +2, okay, zinc ions, okay. So the authors wanted to see maybe that how these ligands are interacting with your p53 protein, and make it deactivate, so it causes cancer or whatever. And here you can find some like experimental data that how they obtain this kind of protein, okay. So the method is X-ray diffraction, and the resolution is this Armstrong, and then you have these values. Later on we discuss these values, that how they are useful in the drug docking, okay. And these are just a little details about the proteins. It's the same as we saw in beginning of this page. So that was all about the PDB. So in PDB, what you do is that PDB is, if you want to summarize in one place, so you can say that the PDB database is used to we use it to find what proteins, okay. So find proteins structure. Okay, not just only structure, but what 3D structure. Okay, so they are in 3 dimensions. They have like the width, they have height, and they have length. So sort of 3 dimension. Okay, so you can find the 3D proteins to for the drug docking. Okay, and another database is the PubChem. Okay, in the PubChem, you don't find the proteins. Okay, but PubChem is useful to obtain the small molecules. So you want to see that if you are using any molecule on this protein, whether it's going to work or not. Okay, so for example, if we were doing this study instead of the authors, so what we would do is that we would like search these ligands, or you can say the small molecules, okay, and we can find in the PubChem. So for example, I just want to see that if this ion is present in the PubChem. So here you write the sulfate ion and do search, and after some time you see that it's giving you some result. So you have different variants and you have to select according to you. Okay, so for example, I'm interested in this. So here is the structure as also mentioned here, but here you can see that they have bit different structure, but overall we can see that it's the same. It's just the representation of the molecule is different, but the structure is same. So in this way, you can obtain the small molecules to bind with the proteins. Okay, but in case if you want to find a new molecule, okay, that is not exist, okay, so for the organic chemist, they always try to synthesize new compounds which can inhibit the cancer. So if you are successful, you can do the in vitro experimentation and you can patent your compound. Okay, so for example, these all compounds are existing. Okay, you cannot patent this compound. So for example, you want to do the new compound, how are you gonna achieve it? Because new compound will not exist in database. So in database, there are compounds that already exist in this world or already exist in the literature, but you want to do something new. Okay, so to do that, what you want to do is that you go again to the RCSB PDB. Okay, here what you're gonna do is that you have to find the option which is called the sketch. Okay, so it should be the PDB sketch. Here we will, or we just write on the Google, maybe it's more easier. So here you go, and you just write, here you just write the PDB sketcher. Okay, so in PDB sketcher, you'll see that there is a tool called the PDB sketcher. So sketcher, sorry. So here you can, in the PDB sketcher, what you do is that you have to put the smiles. Probably you don't know what the smiles are at this point, but we will later discuss it. What are the smiles, okay? And what are the inch keys, or I don't know how to pronounce it, in key or whatever. Okay, so first, suppose that you want to build your own molecule, which you are calling that it's a novel molecule, okay, and it should inhibit the cancer, okay, based on your hypothesis. So what you're going to do is that you should know that what molecules you have, you want to make, okay. So in this case, so these are like the bonds. So this is the single bond, and this is double and triple, and then you have aromatic, and then you have single up. So these are all like the chemist tool until you know these rules, okay. So for example, I want to build one molecule which should contain the like carbon and hydrogen, okay. I want to build like this, and this is just This is just an example, okay? So I want to make the novel molecule, okay? So this is the molecule I made, okay? And here I want to attach the ring. So I want here, the C is the red, means that it's not possible in the real world, okay? So you have to substitute it with this, okay? Maybe we again do this one. So this is our molecule, okay? So this you can say that, okay, this is my novel molecule, okay, which do not exist in the world till now, okay? So I'm gonna use it to see if we synthesize this molecule in the lab, it's gonna inhibit the protein or not, the function of the protein, okay? But now I made the novel molecule, but I don't know that if this exists in the database or not already. Maybe there are some molecules that exist like this. So what I'm gonna do is that I'm gonna copy the ChEMBL, okay? I go to the PubChem, and here I paste it, and I search it, and you see that there are no compound that exist in this database, okay? So maybe you can see that maybe we remove some carbon, maybe we can find something similar. Yes, maybe we found one which is similar to our compound, but we added some more structures, some more rings. So here you can see that this is the one ring, and then you have single bonds. So it's more or less like this, just flipped, okay? You can also flip this one if you want to compare with the PDB, and you can see that this compound is similar to this searched compound. Maybe we have here double bond, we have double bond, we have just these extra bonds here, this one, but well, this was just an example. So that's how you can build your own molecules on these databases, and then you can do the docking. So the summary of this is that we use PDB database to find the proteins. Then we use what? The PubChem protein. Sorry, PubChem database. Okay, so PubChem database is used to obtain what? Single molecules. Sorry, small molecules, or you can say inhibitors, or you can say maybe drugs. But at this point, we don't know if they're going to be drugs or not. If it's working, they are drug, otherwise they are small molecules. Or inhibitor activator is based on the function, that how they are affecting the protein. So here, so in the PubChem, for now we just need these two, okay? But the problems here also arrives is that, sorry, here problem arrives that not all the proteins exist in the PDB database. So for example, if you don't know the protein, okay, oh no, sorry, if you know the protein, but you don't have any structure in your PDB, okay, because no one did the experiment on that, or nobody published their protein. So in that case, we use the Alphafold protein, okay. So Alphafold protein is make the protein like this structure, okay, but with the prediction of the machine, with the artificial intelligence, okay. So it uses artificial intelligence to build the protein structure, which is not confirmed from the experimental, but it's still the author are saying that it's a 90% predicting the well is according to the biological structure, and that's why they won the Nobel Prize last year. So that's really working good. So for example, you said that this protein was never existing over here in the PDB. In the case it was not existed, but you can, so how we can generate this protein from in the Alphafold, okay? So to do that, you need the little programming skills, but right now I'm just telling you the introduction part we're gonna see. So this was the protein, okay, and but imagine that it was not existing in this database, okay. So you had only like this information, maybe this one. Okay. You had this information, just you had only information that you have in your samples, the protein, which is with this code, and it is p53 human. Okay, so you want to dock small ligand on this small molecule on this, but you don't have the structure. So where the structure gonna come from? If it don't available here in the PDB, if it's not available, then you go here. Okay, find your protein, which is according to your analysis, and then you go to the sequence here, and then you can copy this sequence, or you can simply download it. You can do this one, maybe it's better. You copy, and here you paste on this. Since this protein is already exists, so nobody predicted it because it's already existing there. So let's check with other, maybe we remove this one. So here you see that we just paste the sequence of our protein, and here it appears the protein. Okay, so the thing is that this protein comes from the organism or from the artificial intelligence. Okay, so in this case, this protein came from this troglodytes. Okay, maybe you want to see what it is. So this it comes from, so it's come from the real data, not from the predictions itself. Okay, but it matches. Okay, so now you see that you got the protein from here. Okay, but now you have this species which is non-human. Okay, so now you have to search that how much it is similar, our protein, to this protein. Okay, so here it did the homology test for us. So here it is telling the result that it's 100% matching. Okay, so now instead of human, we can also use this protein to do the drug docking. Okay, because we are supposing that this protein is not available in the PDB, so we can also use this. Maybe you will find some sequences that are not 100% identity similar 100%. Maybe they are 70%, maybe they are 80% or 60%, whatever. It depends on what you want to put on your paper or on your results. Okay, there is another protein which is 100% also matching, but here the organism is different. So we have different organism, and again from the same, and the results will keep going. There are many results. You can check any one of them according to your goal. Okay, so let's go till the end. Okay, so we have many results like 101, and here you can see that we went on very last, and here you can see that this is from Monopterus, and here you can see that your sequence matched 72%, and these are gaps. Okay, maybe these gaps will make problem while docking the drug, so you always have to take the more similar one. Okay, so in this way, this Alphafold is useful. If you are not finding the protein in the database, then you can predict the protein based on the computational or already available data from Alphafold. Okay, so this was the small introduction for the Alphafold. Okay, so in the summary, what we do, in summary, we have like this is here we have this is sufficient for the docking, but the problem is that what if the proteins are missing? What if you don't find the protein in the PDB? Okay, then the next option is to use what? Alphafold prediction. Okay, so what you obtain from the Alphafold is the same protein 3D structure for drug docking. Okay, so in the case of you are not finding any protein in PDB, you're going to use the Alphafold prediction. Okay, so this was the database. This is the introduction of these databases. On the next class, we're gonna, not the next class, tomorrow we're gonna publish the video about how to install the system for the docking in the computer, in your own computer, and how to do the Linux part and all that stuff. But on the next video on the courses, you will find that how we can use these databases to obtain our information that we need or we design. Okay, so this is here Alphafold prediction, and we are discuss about the last part, which is the ChEMBL, which called ChEMBL. Okay, so this is not related what we did here. Okay, but this will be useful in same as obtaining the small ligand like PubChem is the alternative of the PubChem. Okay, like there is the alternative of Alphafold with PDB. Okay, if you don't find any structure in the PDB database, you go for Alphafold. In the same way, if you don't find any molecule that are in the PubChem are not present, you can go to ChEMBL. But ChEMBL have one more extra feature, which we're gonna use in our course, which is modeling of the QSAR modeling. Okay, quantitative structure relationship. Okay, we're gonna do on the later classes, but here I just want to show you that we're gonna use these databases in this next class. So if you can explore these databases, would be better for you to get the concept very quickly when we are uploading the videos. Okay, so far we are finishing this class with this database introduction. On the next class, we're gonna install the system for molecular decking, and then we're gonna move again to these databases to do the modeling of the drug. Okay, thank you so much.